Cardiac muscle cells, or cardiac myocytes, differ from skeletal muscle cells in having a long, sustained depolarization. Cardiac muscle cells have a resting membrane potential of around minus 90 millivolts, a threshold of around minus 75, and they reach a peak of about plus 30 millivolts. We're also going to care about what happens at 0 millivolts. In a cardiac muscle cell, the membrane potential is around minus 90 until the gap junctions from a neighboring cardiac myocyte allow sodium into the cell, and they bring the cell to threshold. At that point, voltage-gated sodium channels open, just as in typical skeletal muscle. That brings the membrane rapidly up to plus 30. Whereupon the voltage-gated sodium channels close. At that point, voltage-gated calcium channels open. This is different from skeletal muscle. There's a brief delay, so the potential drops a bit, and then over time, there's a slow decline, and at around 0, they close. At that point, long-delayed voltage-gated potassium channels open. Potassium leaves the cell, and as it leaves the cell, it removes positive charge from the inside, making the inside more and more negative. We go back down to minus 90 and wait for the next influx of sodium to start the next action potential. This period is quite long, so in this case I've drawn an entire beat that is about 1 second long. That would be for a heart that is beating at somewhere around 30 or 40 beats per minute, so a relatively slow heartbeat for a human. Some animals and elephants are much slower, and for other animals, small shrews and rodents, it can have 600 or 700 beats per minute. Let's run through this for the events going on inside a cell. We're starting at minus 90. Gap junctions from a neighboring cell allow sodium into the cell. They take the cell from minus 90 to around minus 75. That positive charge opens voltage-gated sodium channels, and sodium enters the cell. That takes the cell from minus 75 to plus 30. At plus 30, the inactivation gates of voltage-gated sodium channels close, and voltage-gated calcium channels open. Calcium enters. That calcium entry is enough to balance the small leak of charge and keep us close to plus 30, but we switch from plus 30 to plus 20 to plus 10. Hundreds of milliseconds are going by, and finally to 0 millivolts. At 0 millivolts, the voltage-gated calcium channels close. At 0 millivolts, voltage-gated potassium channels open, and potassium leaves the cell. Remember, the equilibrium for potassium equals about minus 100. To get from 0 to minus 100, positive charge would need to leave the cell. Potassium leaves the cell. We go from 0 to minus 20, minus 40, minus 60, minus 80, minus 90. And at minus 90, these channels close, and now we're waiting for the next action potential and sodium entry again to start a new cycle of contraction. The mechanism of contraction is the same one we've been talking about for skeletal muscle in the past. This would be a double strand of actins, but all the binding sites are covered by tropomyosin. The tropomyosin has troponin, and the binding sites are covered until calcium binds. That calcium binding moves the troponin away from the binding sites. Now myosin can bind, and binding triggers the switch from the cocked position to the rigor position. When it does so, phosphate leaves. We move this actin. At that point, the ADP can leave, and it requires a new ATP to release the actin and allow a new cycle to start.